There we go. Close enough. Close enough. No worries.
Do you want any other, introduction?
That's
Other
good.
doctor? Okay.
Professor, doctor, whatever.
Okay.
It's all the same.
Okay. You ready?
Yeah.
Okay, we have Professor Rodolphe Barrangou here with us to talk to us a little bit about what CRISPR is and some of the applications of CRISPR technology.
All right, great. So CRISPR is an actual acronym. All uppercase CRISPR, CRISPR, and it stands for Clustered Regularly Interspaced Short Palindromic Repeats. So they are actually very peculiar and idiosyncratic DNA repeats. They are repeats in DNA that are clustered together at one location in the genome, and they are regularly interspaced with specific periodicity. They are short, usually 31 to 36 nucleotides long, and they're partially palindromic, that's where the P comes from, because the 5' sequence is the reverse complement of the 3' tail, oftentimes GTTTT, reverse complements with an AAAC at the end. So clustered together in the genome, regularly interspaced, short palindromic repeats. And what CRISPR do in nature, in bacteria, actually about half of bacteria, 46% of bacteria, and large majority of archaea, about 90% of archaea, is provide adaptive immunity in prokaryotes. And conceptually, adaptive immunity in eukaryotes is the ability, through a three-dimensional protein-protein interaction, to have antibodies and antigens interact in a lock-and-key mechanism. And this is how our own immune system, in mammals in general, Homo sapiens in particular, enables us to recognize and differentiate self from non-self. And it's protein-protein driven. It's convenient. Proteins are easy to traffic across different tissues and cell types and organs, regardless of the site of infection. In bacteria, we're dealing with one cell. And CRISPR in many ways is the equivalent of antibodies and antigens, but it's DNA driven. And it's DNA encoded in the CRISPR itself. RNA mediated CRISPR RNAs, oftentimes called guide RNAs, nucleic acid targeting, either DNA or RNA depending on the system, but oftentimes it's targeting DNA. DNA encoded, RNA mediated, DNA targeting. And those immune systems enable cells to vaccinate themselves against invasive nucleic acids like bacteriophage, or plasmids, transposons, and invasive nucleic acid by capturing a piece of DNA in between two CRISPR repeats into what's called a spacer. So it captures a piece of DNA in the vaccination process, adaptation process, adaptive immunity. And then once it's captured this piece of DNA, it's about 30 to 32 nucleotides long, 3 turns of a DNA helix. Then it can transcribe this into a small interfering CRISPR RNA, which contains the exact sequence of the immunization event. And that sequence enables the system to guide, that's why it's called a guide RNA, to guide CRISPR-associated sequences called nucleases, Cas nucleases, for sequence-specific targeting and cleavage of DNA. So it's very useful, it's very powerful, it's very potent, it's very flexible, it's very nimble in bacteria as an immune system. But what's really cool about CRISPR is not just what it does in nature, in bacteria, but most importantly, how the molecular machinery from CRISPR-Cas systems has been repurposed as what's called CRISPR technology or CRISPR technologies. And to date, the primary reason why people have heard about CRISPR is because of the use of CRISPR-based machines, actually Cas-based machines, mostly Cas9, the very powerful programmable endonuclease, is to use this CRISPR-Cas technology to drive what's called genome editing. And what genome editing is a pretty simple concept in general. Is you're going to take a piece of double-stranded DNA, like this visual piece of DNA, 2 DNA strands, complementary to one another. And what you're going to do is you're going to cut that DNA. you're going to slice the DNA and create what's called a double-stranded DNA break, a DSB. And you're going to use the endonuclease Cas9 to do so. And you use Cas9 as what I call a molecular scalpel. Okay, it's a very small molecular knife, a small pair of scissors, so to speak, that enables you to cut DNA very precisely and accurately and efficiently and specifically, programmably, obviously. And then once you cut that DNA, right, it's bad for the cell to have a DNA cut. It's very damaging. It can cause disease, it can cause cancers, it can cause death of the cell. So you lose the integrity of your nucleic acid. And essentially what happens is endogenous DNA repair pathways come in, and they're going to repair this DNA, much like you would repair your skin if you accidentally use a razor or a scalpel to cut yourself. You're going to patch it back together. So there's two primary DNA repair pathways that enable the patching of that cleavage right here. One is called NHEJ, non-homologous end joining. When you're going to take those two non-homologous ends that have been cleaved, you're going to patch them back together, sew them back together with like duct tape or fishnet or whatever you want. You're going to patch that back together, but you're going to do so and leave a little scar at the point of cleavage, the point of cutting. That small scar genetically is a small insertion, a small deletion, or a couple of mutations or SNPs, single nucleotide polymorphism, single nucleotide variants in your sequence, and essentially you alter the sequence at the exact point of cleavage. The alternative is to repair this cut more surgically using what's called a DNA template. We have another strand next to it that shows homology upstream and downstream of the cleavage site. And then through homologous recombination, you're going to have homology-directed DNA repair, and you're going to repair that break with a template just beneath it that you can provision as a scientist. And the scientist can design the actual content of that template to specifically generate a particular outcome. It's going to generate a specific insertion. You're going to insert a piece of DNA, insert a gene or more. You can generate a deletion by removing what's in between two homologous regions. Or you can give it a template that has a very designer point single nucleotide mutation if you so wish. Okay, so an HEJ kind of quick and dirty, great for knockouts, out of frame mutations, and very quick editing events. Or more surgical DNA repair using HDR. Importantly, actually, CRISPR only does the cutting itself. The endogenous DNA repair pathways is what generates the edit itself. And the reason it's called editing is because you edit the DNA sequence exactly at the site of cleavage. Okay, so that's genome editing 1.0. In the last few years, people have used different variants and flavors of the CRISPR-Cas machinery to generate different kinds of edits. And rather than just rewrite the text and rewrite words and change typos in genetic diseases, you can actually take a version of this molecular scalpel, this Cas9 that comes in, binds and cuts DNA, and then inactivate it, and essentially render those scissors innocuous. Okay, you shave off the blade, so to speak, and you inactivate them. And what you do when you achieve this is you generate a DNA binding protein that you can locate using the single guide RNA that you design, and you turn those scissors into a DNA binding protein. Now that DNA binding protein can bind DNA with such affinity that it will stop an RNA polymerase in its tracks, thereby preventing transcription. You can even fuse it with a repressor, and they very potently turn off transcription. Or you can tether it to a traditional activator and turn transcription on. Or you can tether it to an effector domain of interest that can change the stereophysical availability or shape or structure of your DNA, helicases and gyrases and others. Or you can tether it to a fluorophore and do imaging. You can do up to multiplex CRISPR rainbow type applications where you can do imaging and location and visualization of your sequence of interest in a cell or in a cell population. Or you can fuse your dCas9, deactivated Cas9, to acetylases and or methyltransferases and then change the epigenetic state of a DNA sequence of interest. And essentially, this is what a lot of people call genome editing 2.0, where you don't just change the wording, you don't just change the writing, but you change the punctuation. With the transcriptome, you can be very loud, or you can be very quiet. You can illuminate it, and you can change the epigenetic state. Essentially, you can rewrite and alter any DNA sequence you want, any way you want, in any organism you want. And people have shown in the last 4 years, since the CRISPR revolution onset, in 2012, 2013, that you can use those CRISPR-Cas-based technologies to alter the genetic sequences, as aforementioned, into any organism of interest. Whether you are a scientist doing genetics on a particular model organism, things as simple as viruses, to bacteria, to yeast, to very complex organisms like animals and mammals, even plants. Or you can implement this to the food ag spectrum, where you're talking about bacteria using the food supply chain. You can talk about animals, you can talk about plants, or you can implement this in medically relevant organisms, all the way to humans, the CRISPR zoo kind of thing. And what you can use is use CRISPR or CRISPR-based technologies to edit the genome and or the transcriptome to understand or model a disease and or to correct a faulty gene version. That's called gene therapy. You're going to deliver a therapeutic agent that is going to rewrite a broken word into your genetic content of a patient. Okay? And what's really powerful is that scientifically we are witnessing a revolution. This is a very disruptive technology. The pace and scale and speed and magnitude at which scientists all over the world in different segments, in different disciplines, in different labs, in different universities are adopting, harnessing, exploiting, implementing, applying this technology is unprecedented. And it's a very disruptive technology because it's very powerful, it's very nimble, it's very quick, it's very affordable, it's programmable, it's multiplexable. There's many things you can do with it. And we are seeing in a very short amount of time, 3.5 years, 4 years, depending on how you count, a very strong impact on the scientific community. And then also we can measure that to the scientific literature, by the pace at which CRISPR papers are being written and or published and or released, or even the pace at which CRISPR patents are being deposited or asked for or sometimes granted to accelerate prosecution by the USPTO. So it's very disruptive scientifically, it's very disruptive technologically, it's very disruptive academically. But beyond the science itself, we're seeing a tremendous impact in terms of the media. And most of you and many students actually have heard of CRISPR outside of the classroom, because when you go on Google, you can get CRISPR news on Twitter every morning. When you look at the written press, journal press, online press, TV, and all kinds of social media, CRISPR is all over. It makes not just the cover of Science and Nature and Cell and Nature Biotech, but it can make the cover of The Economist, it can make the cover of Time magazine. There's CRISPR scientists who are amongst the Time 100 most influential people in the world. You can get a TED Talk on CRISPR. Right? You can hear about CRISPR at South by Southwest. You can get your CRISPR on Yahoo News. You can get CRISPR, obviously Forbes, and Wall Street Journal, and not just the New England Journal of Medicine. So CRISPR is all over. And CRISPR actually has made it far enough mainstream to be on TV. So there's CRISPR in the X-Files, there's CRISPR on Luke Cage, there's going to be shows on TV dedicated to CRISPR, or using CRISPR, or co-opting CRISPR-type-based genome editing for scenario purposes. So the media is all over CRISPR. There's millions of hits when you Google this. And there's hundreds of thousands of videos on YouTube that explain what CRISPR is. This is just the next one. So maybe a good one. But beyond the science and beyond the media, I would argue that in many ways CRISPR has been even more disruptive from a business standpoint. because you see different industries. You see the biotech industry, you see the food industry, you see the ag industry, animal and plants. You see the pharmaceutical industry, the tools and research industries, all flock to this technology at the same time. It's very timely, it's very compelling, it's high potential, it's a safe investment or de-risk investment to some extent, very appealing investment. So we're seeing across different business segments, people from all those industries flock to CRISPR. Whether they're a Fortune 10 company, large cap company, mid cap company, small cap company, private company, startup company, even not-for-profit companies like an Adgene and others. It's very rare to see a technology gather the enthusiasm and the excitement of different types of companies and communities at the same time. And when you look at this from the outside, from a technical standpoint, scientific standpoint, from a media standpoint and business standpoint, it's what I call the democratization of CRISPR. It's been democratized in very short order. And if you look at the data from Adgene, there's 10